Hi, listeners. It's hard to believe, but New Frontiers in Functional Medicine has been going strong since 2015. With over 200 episodes and over 2 million listens, we are growing every year. My goal is to continue to bring you the best minds in functional medicine, epigenetics, and longevity. I want the content to be actionable for the functional medicine clinician and interesting for the rest of us. Thank you for supporting this podcast with your likes, with your honest reviews, with sharing the episodes with your friends and family. And if you haven't done so, please consider leaving a like or leaving us an honest review wherever you've accessed this podcast. Thank you, thank you, thank you to our sponsors. Because of our sponsors, these are companies that I trust, that we vet carefully, that we use in clinical practice, that I use personally. Because of them, everything associated with this podcast is free. If you haven't done so, I suggest you head over to drkarafitzgerald.com to the podcast page where you can access the full transcript for every single one of these 200 episodes. And along with that transcript, you'll get an episode summary. You can get the research citations and additional content that is pertinent to the episode. I myself return to the transcript page all the time to pull out a little snippet of information that I want to recall or access a citation. Or better yet, sign up for our newsletter, again, at drkarafitzgerald.com, and every single podcast episode will drop in your email as soon as it's released. Thanks again. Thank you to our diamond level sponsors, Biotics Research, Rupa Health, and Integrative Therapeutics. For over 40 years, the foundation of Biotics Research has been innovation and quality. Their goals remain unchanged: innovative ideas, carefully researched concepts, and product development with advanced analytical and manufacturing techniques. Biotics nutritional products are of superior quality and effectiveness. They're available exclusively to healthcare providers. Find them at bioticsresearch.com. I also want to give a shout out to our friends over at Rupa Health. They make lab testing easy, fabulous, doable for you, the clinician, and for you, the patient. Consider using Rupa as your super, super, super smart solution to all things laboratory testing. Visit them at rupahealth.com. For over 35 years, patients and clinicians have trusted Integrative Therapeutics for their dedication to quality, potency, and accuracy, and for bringing us exclusive products backed by science. From sourcing of premium raw materials to their meticulous manufacturing processes, they prioritize consistency, efficacy, and safety because our patients deserve nothing less. Find them at integrativepro.com. That's integrativepro.com. Thanks to our gold sponsors, Timeline and OneSkin. Timeline, fueling cellular energy for a lifetime of healthy aging. Find them at timelinenutrition.com. And OneSkin, skin age reversal technology. Find them at oneskin.co. What if we could actually identify and even correct significant autism risk in utero before a baby is even born? This is the promise of the game-changing research of Janine LaSalle, epigeneticist extraordinaire out of UC Davis. With autism rates continuing to increase at alarming rates, 2020 data from the CDC puts it at 1 in 36 kids in the US. New ways to prevent and treat this condition are of utmost importance. Dr. LaSalle is on a mission to change that by investigating something you probably had not heard of yet, the placental methylome. Methylation signatures in the placental methylome are yielding information both for predicting autism in children and for the intervention targets to prevent the development of it. It's incredibly exciting. In our conversation, you'll also learn how placental methylation behaves differently to DNA methylation in other tissues, and how the placental methylome may help us understand cancer, as well as mechanisms that restore a more youthful epigenetic pattern. Hold on to your hats, folks. This is going to be a deep science dive. but know that it's actionable even now. You'll hear how methylation support, specifically in the early pregnancy window, is key, and how it's also protective against insults from environmental toxicants. You're in for a treat with this fascinating conversation that has far-reaching implications. Thank you for joining us. Hi, everybody. Welcome to New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And of course, today is no exception. I am beyond honored and thrilled to be talking to Janine LaSalle. Let me give you her background, and we're going to jump right in to what will no doubt be one of the best conversations we've had on New Frontiers in some time. Dr. LaSalle is professor of microbiology and immunology at UC Davis with expertise in epigenetic mechanisms at the interface of genes and environment. Dr. LaSalle serves as the deputy director of the UC Davis EHSCC, and that is Environmental Health Science Center, and co-director of the Perinatal Origins of Disparities Center. Her lab uses genomic and epigenomic technologies to investigate DNA methylation signatures in autism spectrum disorders and beyond. Her lab utilizes perinatal tissues such as placenta, cord blood, newborn blood spots, and cell-free fetal DNA for epigenetic prediction and prevention of health disparities. Dr. LaSalle, welcome to New Frontiers.
Thank you. Pleasure to be here.
Everybody who's just heard your bio is incredibly excited already. I can tell you, you're just doing some extraordinary work. I mean, I just actually want to, we were talking about how I heard you on the Active Motif podcast. If anybody wants to listen to a really dense scientific discussion of Dr. LaSalle's work, hop over there. It's October 2023, but I heard it at that time and just really was hoping that you would consider joining me. In 2017, I actually interviewed Dr. Moshe Sef, who went on to become a mentor of mine. And in 2017, he said, very extraordinarily to me, that he anticipated us being able to actually diagnose autism in utero. And it was such a remarkable statement to hear in 2017, and something of such promise. I was deeply moved by that. And his suspicion that the interventions might not, that we might actually be able to correct it, in utero before it even presents. And that was that was momentous. It was quite a statement and very moving, and the hope that came with that was huge. And so, when I heard your work, that you in fact were bringing to birth, for to use a good metaphor, like his promise back then just wowed me. And so, of course, we're going to drill into this, but I want to know how you got into epigenetics in the first place. And then, we'll move into talking about how you mapped the placental methylome.
Yeah, so it goes back like 3 decades, I think. So I was a graduate student in immunology. And at the time I knew I wanted to postdoc in the Boston area, but didn't, wanted to maybe explore just outside immunologists. And I met Mark Lalonde at the time, who was at Children's Hospital and Howard Hughes Institute. And it was interesting because we were collaborating with him, and I was cloning T cells, and he wanted to collaborate, and I thought he was going to send a postdoc or a graduate student, but he shows up in the lab next to me in the tissue culture and learning how to clone T cells. And in the process of getting to know him, he is saying, "Oh, I'm looking for a postdoc. Would you consider it?" And then he started telling me about this crazy Prader-Willi-Angelman syndrome. And I, never really liked genetics that much as an immunology graduate student because it was very dry. But I just got fascinated by, okay, how does this occur? And it was just really interesting. So yeah, he converted me into an epigeneticist in about 93, I guess. So, and I remember, at those times when you would go to genetics meetings, it was epigenetics was the quirky exception to the rules. So yes. It was the people working on imprinting, exon activation, and maybe thrown in with like the mitochondrial people, right? The quirky exceptions to Mendelian inheritance.
Like the methyl group on the cytosine was sort of something to be ignored, or is that true?
Like, yeah, well, we had DNA methylation then, but it was not really. Yeah, I mean, we knew that was one of the marks of imprinted regions. That was known about it, even when I started. I was doing more of the microscopy and fluorescence in situ hybridization to look at replication timing at that time. But yeah, methylation was in its infancy. We didn't really have a human genome yet. So it's been just really fun for me over the decades to see the field expand and so many people get excited about epigenetics and how it's not just a quirky exception in a few rare human genetic disorders. It's actually a rule of thumb for most of our probably complex disorders. Yeah.
What a full flip, right? Would you say that that was born out of, I mean, we often talk about it in functional medicine, that this movement towards really lighting on epigenetics was kind of born out of mapping the human genome and then seeing that, whoa, it's not the Rosetta Stone we thought.
Right. Yeah, no, I think that again, I remember hearing those arguments about the human genome and I'm thinking, yeah, they're kind of overselling it. And I think it's exciting because I think it's now people are coming around to the fact that genetics on its own only gets you so far. And we really need to understand that interface, that connection with how our genes respond to the environment. And I like to say, the genome is not in a lockbox away from all the other influences. But a lot of times I think genetics in its silo treats it that way, right? I mean, people will just study the genes alone. And again, that gets you at causality, but we realize that causality is not 100% for most genes. I mean, definitely, and we work on syndromic disorders, which are, close to 100% penetrant, but not completely. And, so even those, there's interactions with environment and we've gotten into looking at Down syndrome, which again is the classic, genetic disorder. We know it's caused by an extra chromosome 21, but yet some kids with Down syndrome have congenital heart defects and some don't. So we've recently looked at an epigenetic signature that can distinguish those. And we're working on Rett syndrome, and Rett syndrome, it can matter, because it's on the X chromosome, you can have identical twins, one that, has what looks like autism and the other who can, is functionally quite well off and going to classes and so you can get this whole spectrum within the same genetic.
Something clearly genetic is not influenced solely by the change in gene. And in fact, the change in gene isn't necessarily a predictor of having outcome.
Well, I mean, it can still be a predictor, but it's not necessarily always.
Yes.
I mean, yes, a child with an extra chromosome 21 is always going to have Down syndrome, but how severe that Down syndrome is can be epigenetics.
You might, I mean, are you familiar with the I feel like Bruce Ames was an author on it, but I don't have it in front of me, and maybe I may be incorrect. What it was giving a multivitamin to Down syndrome individuals demonstrated improvement.
Yeah, so prenatal vitamins, that's something we've worked on in several different projects. And it's interesting because prenatal vitamins are mostly, it's folic acid, which is a major methyl donor in the diet. And it's there for prevention of neural tube defects, and it's been very successful for that. It's one of the most successful public health implementations that's been done. But it also is preventative for Down syndrome, right? I mean, so women that have taken it, but you need to take it in that first month of pregnancy. And then I've been working with Rebecca Schmidt, who's an epidemiologist here, and she showed that taking prenatal vitamins again in that very first month of pregnancy is preventative for autism as well. So we've now started to look at that as one of the variables when we start looking at, when we have a region that is an epigenetic candidate for autism, we'll ask, okay, is that changed with use of the prenatal vitamin in the first month of pregnancy or not? And we've seen a couple loci, yes, where it's very clear cut that there's an association between, and it's always protective.
That's incredibly interesting. And so, listen, there's a lot. We're skipping ahead to like question 6 and 7 here in my list. But I want to ask you, and I want to make sure. So we want to talk about that gene you just identified, because we're talking. Well, we need to get back to the placental methylome, but these genes you're identifying are probably some of the ones that we're going to analyze in utero to determine. And then you've just suggested that you might be able to turn off this genetic involvement by the use of folic acid, or, we always, in the functional medicine space, we're more often prescribing actual activated folate versus the the versus folic acid, the oxidized version. But that's, I mean, same point. We're supporting methylation through a folate vitamin. But I wanted to just throw out there, and then we're going to get back to all these other questions. There was an interesting John Hopkins paper that came out, I want to say maybe 2016, looking at a U curve of serum levels of folate in women who gave birth to kids on the spectrum. So deficiency and excess were both associated. And I just want to get your thoughts on that.
Yeah, no, it's a really good point. Some of that may be the ones that are taking the really high excess could be doing it for medical reasons. So maybe it's that original medical reason, which is why. But I'm also open. We actually are now also collaborating with a group here who is looking at the excess folic acid as well in both a mouse model as well as human organoids. So I think, and it'll be interesting to see what comes out of that work.
We're gonna stay on this. I'm telling my team, we'll follow, we'll stick to you like glue.
Yeah, because it could be both. I mean, and the thing that I've always been struck by, it's really because it's that first month of pregnancy, and at least the data coming out of the cohorts here in California, only about 30% of women were taking a prenatal vitamin in these cohorts in that first month. So there's a lot of room for improvement, right? But because a lot of them have not come for their first prenatal visit yet. So by the time they're once you get beyond that first or second month, you're up to 90% of women are taking the prenatal vitamin. But at that point, yeah, is it more harm than good, or too hard, it's for the wrong people again, and it's a blanket recommendation where maybe what we should be doing is more precision medicine based, right? Because if there's known genetic polymorphisms, this MTHFR TT allele, that if you have that, it's like 15% of the population, they're the ones that really need it the most because they don't metabolize folate as well. But so, yeah, it's like if we could do a screening before to know, what the folate level to give everyone. But I think there is potentially a harm in too much and too late, because we know the protective window is very small, and if you miss it, it might be too late.
God, that's just very interesting. I'm so grateful that you guys are doing this work. Well, in the end, we have folate fortification, so there's exposure there. In non-dairy milks, it's almost like drinking a multivitamin. There's always folic acid present in B12, and it's interesting. And it just makes me think of Randy Girdle's work, and they made huge differences in the agouti mouse gene expression by those early methyl donor exposures. All right, where do I want to go to? Let's first talk about mapping out the placental methylome and what that is and why and how, and we could layer on the placental brain axis and specifically some of the main gene, I think maybe genes that you've identified, the placenta being a surrogate marker of brain, the fact that the methylation patterns are almost in reverse to what we think about in adults. But anyway, let's get into that whole story.
Yeah. I guess you want a little backstory on why we decided to do this, or
yeah.
So it was interesting because at the time, this was around the time when the first maps, and I think it was about 2011, I think was when Ryan Lister from Joe Ecker's lab published some of the first whole genome bisulfite sequencing maps of the human methylome. So, we had the human genome around 2000. This was for me the first. I mean, when I started looking at these maps, it's like, wow, this is really different than and I've been in epigenetics for a while. And one thing that was buried in their paper was this phenomenon called partially methylated domains. And it is like a landscape. If you can think about hills and valleys, if you're looking across a chromosome and the total levels of methylation, there are these valleys that dip down in the overall methylation level. And they'd seen that in a fetal fibroblast line, IMR 90. I was familiar with that paper, and then we had done the first our first whole genome bisulfite sequencing in a cell line model. It was a neuronal cell line, but it's a cancerous cell line. And we're looking at the data going, why is it just so low over we mostly were interested in the imprinted region of 15q, but we're like, why is this whole thing just like dropping down? And then so we got thinking about partially methylated domains, and Diana Schrader, my postdoc at the time, was really a student saying, well, let's try a tissue where it has low global methylation, and that was placenta. So we collaborated with folks here to get placentas, and that's where we basically mapped out that this was, and partially methylated domains had been seen previously in cancer, and in like this fetal fibroblast line. But in most human tissues, if you look at any cells in the blood or most organs in the body, it's really, high methylation is kind of overall, and obviously it dips down over those CPG islands. So it's part of this weird landscape. And then so that brings us to why is it wacky, and why is it what I like to call opposite world, because it's kind of the reverse of what you've probably been trained in methylation, which is methylation is always silencing. So in the placenta, it's true that if you look at CPG islands, ones that are repressed have higher methylation than not. So that fits. But it's when you drop down this whole region, it's the opposite. So now gene bodies, it's like the default is low methylation everywhere. And when the gene starts being expressed, you see higher levels of methylation over the gene body.
In the placental methylome, you see higher methylation.
Higher methylation correlates very nicely with gene expression. It's almost like gene expression leaves these footprints behind in the landscape, which that got us thinking. It's like, well, if there's footprints left behind, could we use that to then look back in time, right? It's a time capsule of what happened in utero. Yeah, so that's how we got into thinking about first, well, just discovering that the placental methylome is different. It looks more like cancer than it does somatic cells. And then also thinking, could this be used potential for, predicting things that, or uncovering things that happened in utero. And so from there, it got us to then thinking about what could we use this for the brain. So in the parallel to all of that going on, we were also starting to do whole genome bisulfite sequencing in autism brain samples, right? So postmortem brain, you don't get a lot of samples, but and there were several studies together that together you could see that there was definitely an epigenetic signature in brain. But obviously postmortem brain has a lot of reasons why it's hard. You don't get much about information at all clinically about these samples. So, and obviously, if you want to try to do any prediction and prevention, you can't sample a child's brain. So there was a whole push in the field to ask, okay, could there be surrogates for brain? And most people looked in blood and seen some things, but blood was never as convincing.
It's been a huge struggle to really put our fingers on anything for diagnostics or prediction. I think you're really kind of the first to come forward with something that could be game changing. But so go ahead, you go.
Yeah. So then again, I'm collaborating here. I'm fortunate to have some great folks here through our Mind Institute, which focuses on neurodevelopmental disorders, as well as our environmental health focus. So we have epidemiologists who started this prospective study. So this is Rebecca Schmidt and Irva Hertz-Pichotto. And so what they did is recruited moms that already have at least one child with autism who are planning the next pregnancy. So now they can look before, the child gets diagnosed with autism, because that second child's got about a 22% chance of having autism. So it's much higher risk. So you can think of it an enriched risk cohort. So we're looking within the family, because if you did this in the general population, it's still this like autism is up to like 2% now, but it would take you a lot to get enough autism samples. And I call this a lesson in patience, because we did wait a while. I mean, it's not an easy study to do for them. It took, 10 years of collecting samples before we really had enough to even, we did it one study earlier to just kind of proof of principle to see that we could do it. And then in 2022, we published the larger study where we had a replication group. There's a similar group at Hopkins. Danny Fallon's group had a similar type study, so we could do a replication with that.
And this was Genome Biology, I believe you published in, and I think it's a free full text. We'll link to it on the show notes, you guys.
Yeah, so that was exciting because it was like a long time in coming. And yeah, the second paper when we had enough there and we could do the replication, we really had a very strong signal on chromosome 22. And it was in this region where there was really not no known genes anyhow. It's what I call these sort of the off the map regions of the human genome. And it turns out there was a little transcript in there. It hadn't been characterized. It's one of these that has a LOC with a bunch of numbers after it. So it's not a well annotated by any means. So we did a little functional,
Wow.
tried functional studies to figure out, okay, what is this? Does it make it's been annotated as a non-coding RNA. But we were curious about whether there may be a peptide involved. So my postdoc at the time, Antonio Gomez, was very clever about saying, okay, let's go in and find the most predictive peptide and design an experiment to look for that. So we were able to show that this gene, which we eventually named NHIP for neuronal hypoxia inducible placenta associated, it turns out that it makes a 20 amino acid peptide that all we know this now, it goes to the nucleus. We are currently working on it. We've got a new grant to understand its potential. It's hard when you get something brand new because there are no reagents to help.
Right.
So we have to develop all those ourselves. But the
Incredible.
promise is that it looks like it's neuroprotective from the evidence from that paper because it was lower in autism. The methylation was lower and the expression was lower in both placenta and the postmortem brain we saw was lower, suggesting that this is something that is induced with oxidative stress or hypoxia during pregnancy. And if you inherit the genetic predisposition to not express as much of it, then that is what that genetic predisposition was also associated with the risk for autism in the small cohort. But obviously we need to do more to figure out if this is going to be a more general diagnostic or therapy for autism.
So, and this is where folate would be obviously successful. So you're identifying this basically unknown gene. It was sort of placed in the junk region. Is that right?
Well,
Sort of like,
it's a very repetitive region of the genome. So a lot of times it's left off the maps of array. It's not well represented on either the methylation arrays or the SNP arrays. So yeah,
wow.
it wouldn't have probably been seen by other approaches.
Yeah, minor details, huh? When we just ignore minor details. So you discovered that this was associated with hypoxia, and paradoxically, it's hypomethylated and therefore inhibited in individuals with higher risk for autism in this cohort that you were looking at.
Right. Yeah. And
And
there's also a genetic, yeah, I didn't cover that yet, but we found, because it was such a striking thing. I'm like, well, maybe there's something genetic going by too, because it really it's rare that you see something that large. And so we did find there's a insertion, a 1.7 kb, this extra chunk on the chromosome upstream of the gene. And that seems to, yeah, so if the child inherits 2 copies of that insertion, that's the risk for autism. But we did see some in the typical group that had that, but they were more likely that their mothers took the prenatal vitamin.
good.
So,
Okay, that's where I
and
was
our
going
numbers
with
are really
that.
low, but it suggests that taking, even with the genetic risk, which is pretty common in the population, we're still, figuring out but it's actually in some of these larger databases, and it's about 70% of people have at least one allele of this insertion, and probably 10 to 15% have 2 copies. But again, it's kind of the similar numbers as that MTHFR genetic polymorphism that I mentioned. So if you take the prenatal vitamin, you can overcome that genetic risk in many cases.
Okay. All right. So how did you guys figure out it had to do with hypoxia?
Yeah. It was mostly we were looking at where is it expressed in other cell lines, and what was remarkable is we have this cell line model. It's called LUMIs. It's a neural precursor cell that you can induce to become neuronal. And when we looked at sort of the undifferentiated versus the neuronal differentiation, we saw NHIP increased, and so it's associated with neuronal differentiation. But we were also looking at the gene list, we also overexpressed it, and we're looking at all the genes that were overexpressed, and hypoxia came up in that. So the combination of the neuronal differentiation, which already kind of induces oxidative stress anyhow, because the neuron's becoming much more metabolically, well, it's changing very, very rapidly metabolically. So then we decided to try this hypoxia mimetic, which is cobalt chloride. And interestingly, the differentiated neurons were responsive to that they became increased reactive oxygen species in response to that. And so that's the question we're asking: is NHIP maybe protective? It's right now it's correlated with it. We're asking, is it going to be protective?
Right. So actually being able to have it on doing its thing, because there's lower incidence of autism, it's quite possible.
Yeah, and if we could add it back as a peptide, that would be ideal. But it also could be added back as a gene as well.
Say that again. It could be edited.
No, added like
added.
therapeutically used as a peptide, or
prescribe a peptide in moms who are pregnant, or would you do it pre-pregnant, preconception? I mean,
yeah, these are all good questions. Yeah, I think we could identify moms at risk and think about what, this is a potential therapy. It's down the road. We obviously have more work to do, but
yeah. Okay. All right. I've got a few questions from this though. So you identified this placental gene and you've named it NHIP, and will you be able to, is this something that we'll be able to use to take a placental specimen? I mean, what would be the ideal,
Yeah, that's where we're thinking potentially the cell-free fetal DNA. And so far we've published on this in a macaque model of rhesus macaques. And this was part of a collaborative group where we were looking at the effect of maternal obesity. It's a known risk factor for autism, ranging from about 1.6 to twofold higher incidence of autism. Like for severe obesity, it gets up to about twofold increased risk. And this was interesting because we have a primate center here, and there are naturally like a lot of the animals in captivity, like humans, are getting obese, including the rhesus macaques in the colony. So they just took ones that were already sort of higher body weight obesity in the macaques compared to the lean, and they actually were able to do two different interventions as well. And this is headed up by Cheryl Walker here, who's an obstetrician. And so they did a statin drug versus a calorie restriction. And yeah, numbers are really low, again, for macaque studies, much even lower than human studies. But it was interesting because we were able to use the methylome to then go and look for changes that were with these four groups. And again, we found some big regions. NHIP was there because the interesting thing about NHIP is it's primate specific. So it wasn't one of the ones, and I've looked in that locus. I mean, again, it's not annotated, but I've looked in that locus, and that one was there, also showing up in this model. But we found two other regions that, again, the primate genomes are even less well annotated as the human. So we found a region that's not well known, although there's an miRNA there that was interesting, and probably some host genes for that miRNA. And then we found this whole DUX4 group of genes that usually work together, that were there. And the interesting thing, I think for the clinical side is, yes, it's the animal model, but when we then take those methylation regions, we compared it back to like the placentas and the brains that we've done before, those epigenetic signatures, there was a significant overlap with the human autism samples.
This episode is supported by Practice Better, a leading all-in-one practice management platform. Most health and wellness professionals love what they do, but feel overwhelmed with time-consuming administrative tasks. With Practice Better, you can automate tasks like onboarding, scheduling, charting, nutrition planning, and billing to help you better focus on the parts of your practice you love. New Frontiers listeners get 20% off the first 4 months of any paid plan at practicebetter.io with code KF20. Visit the podcast page on drkarafitzgerald.com for more information.
As we go deeper into the world of longevity science, I want to introduce you to a company rewriting the rules of skincare. Founded by 4 PhDs in skin science, OneSkin has developed a peptide scientifically proven to address cellular aging. By targeting senescent cells, their OS01 peptide helps skin look, feel, and behave younger, visibly reducing lines and wrinkles and improving skin's texture and overall appearance. To learn more about their groundbreaking approach, visit oneskin.co. That's ONESKIN.co.
So a lot of times we go back and forth between the human and the animal models. We usually like to start with the human and then try to have an animal model that models that human condition. And then we try to bring that animal model, what we find from the animal model and compare it back to
Interesting.
human signature. And in this case, yeah, we definitely got a strong signature that overlapped with the human autism and other neurodevelopmental disorders.
So NHIP as a piece, but you're expanding it to other regions.
Yes.
Did I
Yeah.
get?
So I guess, sorry, I got off the track of the cell-free DNA. So the story behind that is actually they were trying, so the thing about monkeys is it's really hard to get their placentas, at birth like we would with a human, because they usually, you leave them alone at birth. It's an important part of the birth process that they don't get separated from their infant. Otherwise, they might reject the infant later. So we couldn't get, the term placenta. They had actually done, Cheryl what got in done the equivalent of CVS for the monkeys to get placental biopsies, but we didn't actually, for some reason, it's a stringy tissue and it was really hard to get DNA out of that. But what they did take was blood throughout the pregnancy, four time points. And they were already doing cell-free DNA because they wanted only male births. Again, monkey studies are really expensive and hard, so they only wanted males. So they were screening for Y chromosome DNA to confirm that they were male births. So we were able to get that cell-free DNA, which includes the fetal cell-free DNA, and that's where we really saw these patterns. It's a cell-free DNA from that. So, and what was remarkable actually is these regions that we found in the cell-free DNA often very much match what we saw in the brain. So it's like the placenta cell-free DNA was predictive of the brain. We also had behavioral, the nice thing about the monkeys is that you can then follow them up and do behavior. So there was at least the social behavior that they were measuring was correlated then with this.
The genes that you guys were seeing, what you were picking up. Oh, so then cell-free DNA will have some fetal DNA and you can differentiate that in the laboratory. And that would be a much easier collection than a placental biopsy, clearly.
Yeah, if you wanted to be able to tell during pregnancy what the risk is. And then I think that's an interesting idea. Yeah.
Well, I mean, where is that in development? When are clinicians going to have access to that?
Yeah, well, I mean, we're a little aware. We are doing this now, going back to this Marble study, which I was telling you about, the prospective study. We're going back to the cell-free to do cell-free fetal DNA, the ones we had placentas from. So we have that in progress. I'm a little wary of pushing this too much because, we also have the neurodiversity community that will say, wait a minute, you're trying to, yeah, I don't want to, I mean, it's because wary of also what's happened with Down syndrome and the genetic test. So I think it needs to be clear that this is not a genetic test and it probably will never be 100% predictive. But if we could do an intervention during pregnancy to help, again, the maternal environment but again, that's the question. I think we need to have a clinically actionable treatment for it.
Well, I mean, clearly, number one, as you've already said, would be, adequate methylation support, or, as you guys are continuing to investigate, whether in some cases there might be too much. But the other thing that you have looked at in your work are toxin exposures. In fact, you talk about this methylation, this very unique placental landscape, these imprints. Maybe the imprints not the best word, but these methylation signatures are like reading the book through time, like seeing the environmental exposures over the life of the placenta. So it's what the baby's been, the fetus has been exposed to. And I think I might be able to say that there are toxins are influencing this landscape, correct?
Yes, yeah. And so we've looked primarily so far at PCBs. So these are chemicals that, were a use starting in the 30s, 40s, 50s. They were banned in the mid 70s, but because they're called persistent organic pollutants for a reason, is they don't break down very easily in the environment. They are lipophilic, so they bind to lipids and they bioaccumulate up the food chain. And of course, humans are at the top of the food chain. So we still, although there's a decline in the environment over time, and actually in our study, we were using the marbles cohort again. We could see one of the best predictors of the amount of PCB in the maternal blood was year of birth, their age, or combination of their age and year of birth. But so it's decreasing. But then when we correlated the PCB levels to the methylation levels in the placenta, we did see some groups of genes that were or regions. Actually, what we ended up finding were sort of some of these partially methylated domains over genes that were also observed in autism. So the CSMD1 was one that really came out. AUTS2, which is another autism gene. Those were the two that came out. And interestingly, we also were collaborating with another group, Pam Lyne's lab, who is a toxicologist, and she developed, so again, we go from the human, we want to then model it in a mouse. So what she did was design a mouse model of PCB exposures based on the levels and the combination. So there's multiple congeners of PCBs, it's a different mixture. So they took the same mixture of PCBs that were found in the marbles cohort, which is a high risk autism cohort. So moms that have a high risk of having a child with autism, we model that in the mouse, and then we looked at the placenta and brain in mouse. And remarkably, those two genes came up in the same thing, the CSMB1 and the and AUTS2. So it gives you some confidence that we are finding sort of these epigenetically susceptible genes that when we look across species, we still see them.
So I guess I have two questions. Through the functional medicine lens, so early in my career, I was in a clinical laboratory and we were looking at the methylation cycle. We were looking at glutathione and the amino acids involved in that, and S-adenosyl methionine and S-adenosyl homocysteine and oxidative stress like 8-hydroxy-2-deoxyguanosine and lipid peroxidation and so on and so forth. And what was interesting, I did this itty bitty data dive looking at a cohort of individuals with kids with autism and their homocysteine status, just as homocysteine being sort of like the surrogate marker of what's happening with methylation. And they were and it was low compared to what we anticipated in a normal in a neurotypical cohort. And that's pretty routine, back when I was in the lab, that you would see imbalances in methylation and sulfuration and glutathione status, sort of allowing oxidative stress to be kind of uncontrolled or insufficiently controlled. And that was demonstrated and published on, Jill James.
Jill James, I was going to say, she's talking a lot on that. Yeah.
Yeah. And does that fit with what you're talking about, and perhaps why a prenatal? Just thinking beyond folate's a piece of it, but B12 is there, and maybe there's a little bit of choline because it's prenatal. And anyway, I'm just curious.
I think the prenatal vitamins are counteracting a lot of different. There's probably multiple exposures and multiple genetic contributions too, which may not be 100% penetrant if you actually had proper nutrition as well. So
offsetting the PCB damage and the myriad other toxins that you'll uncover, I'm sure, over time.
Yeah, and we are looking at that in the mouse model of the PCB exposure to look at folate as a potentially protector of that. So
that's fascinating. And so back then, this was early in my career, it looked like the incidence of autism was just rising meteorically from, very low incidence in the 80s, to, I think we must be probably at 1 in 5 boys now, or something really pretty high at this
One in
point.
5 boys now, according to CDC. Yes.
And so why, why is that? What do you think?
It's a very difficult question to answer, but I agree with you. I mean, again, and it was interesting because I wasn't necessarily working on autism. I came here in the late 90s, and that's about the time the Mind Institute started in Sacramento. And so I was going to a lot of the talks on, what's nice is there was a lot of different disciplines coming together to think about autism. And I remember seeing these, numbers going up at that time, and the epidemiologist would say, well, we can't know because the diagnosis has changed every time a new DSM comes out, the diagnosis changes again. So you really can't go backwards and ask the same question because the diagnosis and that's really, I guess, been one of the motivating factors for us too, is can we come up with a molecular test for at least something else to go along with just the behavioral diagnosis, because there are biases. Yeah, if you look over time, girls, I think, were really underdiagnosed previously. It's getting better, but they're still probably underdiagnosed compared to boys. So, I do think that there's a biological reason why there's more boys, and it's about a 3-to-1 ratio. But yeah, we actually are looking now at this historical cohort. What we did is go back from blood spots to in this, I don't know if I have time to get into that cohort, but another cohort where we could go from blood spots that newborn blood spots that from kids that had been diagnosed, and their years of birth starting from about 1989 to 2014. And we can see even in that data set, there's almost a different epigenetic signature between the two. I shouldn't maybe talk about this too much because the paper's close to coming out, but so I guess I just say that we are seeing it in the data, that the diagnosis has changed over time, and it makes it very difficult to say that autism diagnoses from different decades are the same thing biologically. And you think about across the world too, is like, how do we even know the real prevalence of autism in other countries if it's the diagnosis, a lot of kids may not be getting diagnosed. There's a lot of health disparities in that space too. If you want me to get into that, but I mean, can you see it again in the California data that the kids with autism are more likely to be sort of higher socioeconomic compared to the ones that are that would be maybe diagnosed with developmental disorders, disability, DD instead of ASD, and you can see that in the data. So it's frustrating as a researcher because you
just lack of a precision diagnosis.
Lack of precision, yes. But
Yeah.
I mean that's probably true of a lot of diagnoses in medicine. So I think that but I think it's something everyone needs to face, and I think researchers especially need to be aware that just because your samples say autism, that may be different based on. You really do need to pay attention to what the criteria for diagnoses were and when it was done, because it's changing over time.
Right. Yeah. So certainly having a molecular diagnostic will move things forward.
Yeah. What I like about the prospective
look.
study we've done is that all of those kids were diagnosed at the same place. They're all done at the Mind Institutes, and they go through the typical kids too. And what's interesting is in those, I said it's about 22% that have autism. Only about 50% are completely typical on all measures. And there's this other category that we call non-typical development that is somewhere in between. And they're the ones, that have learning difficulties or probably ADHD. It's kind of that other category. So again, the whole spectrum of neurodevelopmental disorders is larger than just autism. And again, it seems to be the higher risk for autism goes along with higher risk for these other disorders as well.
It'll be interesting as you peel that back over the years, what you're seeing with these molecular signatures.
Yes. Yeah, I know. Because I think it is, we might be able to get at some of the disparities in
yes.
Yeah.
Well, and concurrently, I mean, one of the reasons people migrate to physicians migrate to functional medicine is because we're so moved by having a sense of what the mechanism is, because that can inform our clinical decision making. So just the name of the hypoxia inducible gene, suggests the solution might be, supporting antioxidant status, and just linking that back to what we know about glutathione and, and that just walks hand in hand with the fact that, we've got a toxic burden, all of us. It's unmistakable, and it does seem to be increasing. And to your point, the persistent organic ones are hanging around, plus we're layering on,
Yeah, well, now we have
additional.
PFAS, which is the other
Yeah.
chemical, and
Yeah.
oh yeah, we have a group here at Davis who's very interested in wildfire smoke exposure because we're in California, and we just had some really bad years of wildfire smoke exposure, and especially in pregnancy, it's probably all the combination of things we know about air pollution,
Yes.
about smoking, but you get all
Right.
kinds of chemicals delivered through
Well,
smoke.
and some of the pretty scary stuff around glyphosate and some of the I guess the cancer and neurological changes. All right, let's see what else I want to ask you here. So I just want to go back to I know there's a couple questions, and one hopefully I'll pull back here in a second, but just thinking about the methylome, the placental methylome, I want to nail down on why this hypermethylation, the hypermethylation signature is suggestive not of active gene transcription, but of gene transcription in the past. I mean, can you just speak? So why does the placental methylome A look like cancer, and and
Oh
B,
gosh, okay.
I guess both.
There's a lot to unpack. Let me think.
Let me say this. People listening may have we know tumor suppressor gene hypermethylation is a thing in cancer. So there's an understanding of that here, and that the oncogenes can actually be on. So there's this paradoxical occurrence in the tumor microenvironment of hypermethylation of protective genes, hypomethylation of damaging genes in the cancer microenvironment. Actually, incidentally, that's in the aging the methylome as well.
Right.
But then you're seeing it in the placental methylome. So I want to understand that. And I'm just, and then layer on why hypermethylation is suggestive of activation. Because anybody who's been listening to this podcast and, following our conversations here on epigenetics, I tell everybody to, just imagine those red lollipops, right? And they inhibit transcription. Just structurally, you can't get in there. The red lollipops being the methyl groups on, on the CPGs on the cytosine, and you can't dock. So when I was reading your work, I was completely struck by that, because
I know I literally had to go back and I changed on my teaching slides. I think after I saw the whole methylome landscape, and I'm like, this is not this simple anymore. And,
and you know what? There's hints of that in the literature. So I would read in the literature, and there's like a sentence of,
I mean, even back in the days for the Prader-Willi locus, when we were doing these things honestly, I did Southern blots to look at DNA methylation in those days. And there were ones that were the opposite, right? We definitely had the one at the imprinted control region, which was always the maternal, but then you had these other regions that were the opposite. And now that we have the whole view, we see it. And so this is a pattern. I'll get back to the placenta and cancer, but let me just cover this first, because, you see it on the inactive X chromosome, and you see it on imprinted regions where there's this big block of heterochromatin on one chromosome.
Can you define imprinted regions?
Okay,
please?
yes.
Yeah.
So imprinted regions are where there's a difference between it's inherited from the maternal or paternal chromosome. So for instance, in Prader-Willi and Angelman syndrome, you can have the same deletion on 15q11 to 13, but Prader-Willi syndrome would have that deletion inherited from the paternal, and Angelman would have it deleted from the maternal. And it's because there are differences in gene expression across these regions. But it's this whole block of a chromosome, right, that has an imprint. And like the inactive X chromosome is like that in females, that in every cell, you've got one of those cells that's completely turned off, and it turns into like this block of heterochromatin called a bar body. So there are two examples of these big blocks of one of your two alleles is getting turned off, right, as a whole chromosome. And when you see that, those regions actually have lower methylation. And that's been seen for the inactive X chromosome. We've seen it for sure in the imprinted regions. So it's acting like, that partially methylated domain. So I really think it's the lack of accessibility to the DNA methyltransferases, which is why you're not getting the methylation in those regions. But again, going back to, so where else do we see this? And the placenta, yes, but I'm going to go back even earlier before the placenta, because we also did this to ask, okay, why is it in placenta? And if you go back to the oocyte actually looks like this as well. And it's interesting, you have the immature oocyte, and when it becomes mature, there's a whole gain of methylation. It increases methylation, but it always increases over gene bodies. So what the placenta is really more like oocyte. And also too, if you look at there's not a lot of data in human, but there's some of the blastocyst, the different stages of you can get some blastocyst cultures, and the same thing there. You see partially methylated domains in oocyte, those pre-implantation embryos. And then once it's implanted, it's implanted, you think about, so the trophectoderm is that layer of cells, that if you see a picture of a blastocyst and implantation, you've got the inner cell mass, which becomes the embryo. You get that circle of cells around it, that is the trophectoderm. Trophectoderm becomes the placenta, and the inner cell mass becomes the embryo. So the trophectoderm keeps that pattern that was there in early pre-implantation life, whereas the embryo itself gets highly methylated right after implantation. So why does cancer, I would flip the question, I guess, because I think we shouldn't be asking, why does placenta look like cancer? We should be asking, why does cancer look like placenta? Why does aging look like placenta? I think they're, developmental origins, and it's going back. Why create a new program if you can go back and use a program that you've already used? It's erasing some of those somatic marks and going back to an earlier stage. Or, that's one possibility. The other possibility is you still have, if it really is developmental origins of something like cancer, maybe because of the stress in pregnancy, you didn't quite finish the program and all the cells. And all it would take is like a couple of cells in there later on, because they have that earlier epigenome marks, they are more likely to turn into a cancer.
That's pretty fascinating. Yeah. So in cancer, it could be regressing to a more primitive, less controlled state.
Right. And primitive is interesting too, because if you think about all mammals have pretty high levels of methylation, but if you go to vertebrates before mammals, they actually look more like the placenta in that, overall lower levels of methylation. And when you do see methylation, it tends to be over gene bodies. So like zebrafish, xenopus, organisms like that have methylation patterns that look more like the pre-implantation and placental state. So it's going back not only to an earlier developmental state, but also,
that's pretty interesting.
More primitive state, as you call it. Yes.
And aging looks like this as well. And why is that?
I unpack aging. Yeah, there's definitely strong epigenetic signatures of aging. That's clear. And they're different from what you see at like telomere, So, is it the result of, all accumulated oxidative stress over time? Are we overall eroding some of those programs that we put on and going back to that earlier stage? Yeah, I think these are all really fascinating questions. I haven't really
got your area
field, but no, I follow it, but it's it's fascinating. But I think a lot of it can come back to, again, those developmental origins. We tend to think of, this any disease that occurs later on, you think, oh, it must be something I did, in recent life, but sometimes it goes back to, in utero, or potentially even grandparents. So,
yeah.
Well, think about generational impacts.
Yes, that's right. Fetal origins of disease, or, things like the overcalyx cohort or Dutch hunger winter, where we can see generational influences on the methylome from, those exposures, maybe starvation or insufficient food or excess. And then you see that handed down in the development of certain diseases like cardiovascular disease and diabetes. Well, Sinclair's lab, and I know we're at time, but I just want to ask you, David Sinclair at Harvard, and, they're looking at the aging phenomenon in mice, in an optic neuropathy, age-related optic neuropathy, and just aging in general. And, he describes it as, kind of being able to drive age forward and then kind of reverse it, that a youthful pattern actually remains at some level in the epigenome. even as those aging changes happened. And that seems to be in keeping with what you said. And then I don't know if you're familiar, somebody who podcasted with me recently and was again another tour de force podcast was Victoria Sebastiano out of Stanford. And he developed a clock. He's doing cellular regeneration. So they're using Yamanaka factors to really turn back the hands of time on cells. And then he developed this bio clock that incorporates an epigenetic bioage clock that incorporates the entire methylome and finds that the changes that really radically alter all of the hallmark of aging are measured on the polycomb genes, I think. So can you speak to that and define polycomb too?
Yeah, so polycomb regions are ones that they tend to be developmental transcription factors. But again, they're part of that landscape of the methylome. You can see them because they always drop down to they're kind of similar to partially methylated domains, but they tend to be smaller. And instead of partially methylated domains are very tissue specific, like when you do have different tissues, whereas the polycombs are in every tissue you look at, you see this drop down. And what's interesting too, genomically, they're like, we call them archipelagos of CPG islands, because instead of having, one gene with one CPG island, it's almost a cluster of higher CG content, which again, I think evolutionarily that's interesting because they are more, again, leftovers of earlier evolution when we did, like mammals basically have gotten rid of a lot of CG sites because of their, the expense of having a genome with high methylation is you lose CPG sites. And that's why we have CPG islands in the mammalian genome. But these polycomb regions are almost like remnants of past evolution, that they're very well conserved. So these are developmental transcription factors that, we've had that are probably the most conserved genes in our genome across organisms. And their epigenetics is different. They don't use methylation in the same way. They tend to use the polycomb modifications on histones, so H3K27 trimethyl. So that's where you get these bivalency of histone marks. You can have both an active and an inactive mark in the same region. And it's really interesting because we think in placenta, in these regions that are lower methylation, they're actually using more of the polycomb marks compared to later on. And that's also, again, shared similarities with cancer, where it's using the polycomb, this bivalency, this sort of poise. It's a poised state where the gene's trying to decide, should I be on or should I be off? But it can go either way. And I think it's the plasticity of that state which contributes to the plasticity of cancer, the plasticity of placenta. Yeah, and I did want to say too, placenta is like a cancer, right? So what it's doing is going out and grabbing those blood vessels. It's doing angiogenesis. It's bringing in blood supply to the fetus. So it's functionally very similar to cancer.
It's really extraordinary. I mean, it seems like we're at the precipice, although we'll no doubt be here for a long time, really penetrating some serious mysteries. Linking the aging to the earliest developmental, chain happenings, and from an evolutionary thread, pulling that forward. I mean, it's absolutely mind blowing. And I just, again, appreciate you for being willing to spend some time with us. What I was going to say earlier that I was trying to pull out of my brain was just that Dr. LaSalle provided me with a really lovely bibliography. And I know certainly a number of these are free full text. So if people want to look at the PCBs research that she's done, the RET, the NHIP, gene discovery journey, and folate, turning that on, like all of those papers, links will be in our show notes to her full bibliography. Maybe we'll pop the journal imprintone conversation in there so that you can, refresh your mind on what that's about. And we'll put the Sebastiano conversation in there as well. Maybe even Sinclair, I podcasted with him quite a while ago when they were just starting to work on this, so that, if you want to do an epigenetic deep dive, we'll make that available to you on our show notes. I'm looking, I was going to ask you about sex differences. We touched on that a little bit, but we'll park her, again, her bibliography there. I'll just continue to follow you as, some of these things come forward. The peptide that you talked about possibly being able to be prescribed early in pregnancy, and turning around some of the development of these, neurological disorders, and then your continued work in collaboration, looking at toxins and looking at vitamins and so forth. Thanks again so much. And if there's anything I missed, you wanted to
No, I
jump
think you
in.
covered a lot. So
We covered a lot.
hopefully it's useful.
Yeah. Janine, thanks for joining me on New Frontiers.
Okay, thanks so much.
As always, thank you for listening to New Frontiers in Functional Medicine, where our sponsors help bring the very best minds in functional medicine. And of course, today is no exception. Not everybody can be a sponsor on my platform. I appreciate the good work, the relentless research, and the generous support from my friends at Rupa Health, Biotics, Integrative Therapeutics, Timeline, and OneSkin. These are brands I know and trust and use in my clinic and can confidently recommend to you. Please visit them online and let them know you learned about them on New Frontiers. And if it's not too much to ask, consider leaving a thumbs up and a kind review wherever you're listening to New Frontiers. Thanks.